Today we will continue our discussion of this biomimetic chip that replicates the pancreatic cancer migrations and which has been validated by in vivo mouse studies in our previous videos. So in this video we will continue to understand why these migrations and this ablation of these endothelial cells occurs. So one of the hypotheses that these researchers have tested is that whether this TGF-beta is uh, promoted the ablation of these endothelial cells by the pancreatic cancers. So what they did is they did a two experiment. In the control experiment, they inject DMSO in the biometric blood vessels, and in the actual experiments, they inject this SB431542, which is the inhibitor for this TGF beta, uh, and they have started injecting after 7 days of this kind of this pancreatic cancer migrations. That is when they reach the biomimetic blood vessels. And what they have observed that this when they use this SB431542, they can see the decrease in the ablation of these endothelial cells compared to the control case, that is DMSO. And when they quantify these images, they can see that percentage of the replaced vessels area by this pancreatic tumors is very high in the case of DMSO and are significantly lower when they use SB431542 reagent, which is the inhibitor for TGF-beta. And when they check this phosphorylated as SMAD2, which is the one of the signaling factor for this whenever this TGF-beta gets activated, and this experiment did in the 2D cell culture, and they can see that the phosphorylation SMAD2 is reduced when they use this SB431542, both in the case of these endothelial cells, also in the case of this PD7591. We can see the decrease in the SB431542 compared to the DMSO case. So this suggested that it's the TGF-beta which is playing the important role towards this ablation of this endothelial cells by the pancreatic tumor cells. And next, after checking these biomimetic results, they decided to do in vivo studies on the mouse, and they did the similar experiment. That is, they in one group they injected DMSO, in another group they have injected this SB4 5315 and they checked the mice after 1 week, and also they checked some other mice after 2 weeks, or they sacrifice the mice after 1 week and after 2 weeks in different populations of mice, and they can see that there is an increase in the endothelial cells density when the cells are treated with SB4531542, both in the case of 1 week and 2 week, and I do not see too much increase in the proliferation of the endothelial cells between 1 week and 2 week. And this thing they also validated using this marker, that is Ki67, and that data is in the supplementary figure. You can refer to the reference link and check the supplementary data for that. And next, what they did is they also check the apoptotic factors that are present in the control case versus the this actual this reagent, that is TGF-beta inhibitor reagent, and they can see the apoptotic factors also reduced compared to this week 1 to the week 2. However, these apoptotic factors increased in the case of when control case, that is DMSO. So this also indicates that in the control case there is a continuous ablation of the endothelial cells, and whereas in the case of where there is a TGF-beta inhibitor is used, they can see the decrease in the pro-apoptotic factors. However, when they compare between week 1 and week 2, they can see the increase in the pro-apoptotic factors, and also they also check the tumor volume and those data is the supplementary data, supplementary figure. And they also see the increase in the tumor volume. There is also see there is that there is no change in the tumor volume when they compare between DMSO control case and these TGF-beta inhibitors. However, this TGF-beta leads to the decrease in the ablation of the endothelial cells, but it does not affect the growth of the tumors. And these final figures is about this week 1 and week 2 of the mouse models, and they can see the apoptotic factors in the case of this control case compared to the control case, and which is that is compared to this TGF beta inhibitor case. They can see that apoptotic factors are reduced in both the cases. So this indicates that although the TGF-beta plays a role in decreasing the ablation of the endothelial cells, it does not affect too much on this tumor growth, as the tumor growth between this control case and the this SB431542 reagent case are almost similar, and those data are available in the supplementary figure. If you need to understand this experiment in details, or need to understand all the different dimensions of this paper, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.